Where Old Harry waits at sea, if you look west over Swanage, Durlston with its castle sits on the south-eastern tip of the Isle of Purbeck. Limestone cliffs rise from the ocean, giving way to hay meadows and calcareous grassland managed sympathetically with grazing since 1970. This study seeks to identify whether Durlston Country Park's diverse meadows feature far greater species richness and wildflower abundance in contrast to summer grazed, short sward habitat. The purpose being to decide upon ideal management practice for increased biodiversity, or whether both habitats coexisting as a mosaic confer greater benefits for wildlife. With much of Britain's unimproved grassland and hay meadows lost to the plough, chemical fertilisers and improved growth of silage, a hugely important habitat has been in rapid decline since World War II. Once a common sight, traditionally managed hay meadows contain an abundance of flowering plants, providing habitat for reptiles, ground nesting birds, feeding bats, and when harvested, a nutritious winter food source for livestock. Moths, butterflies and other invertebrates thrive with flowering plants, often laying monophyletic larvae that depend on one species, such as the small blue butterfly's relationship with kidney vetch. Livestock is removed from the meadow in March to allow plants to bloom. By midsummer, a diverse array of tall grasses and wildflowers can be seen, later setting seed providing protein in next year's progeny. Hay is cut in July or September and allowed to lay where it lands, drying, providing food and shelter for mammals, birds and invertebrates before being baled or stacked and stored for the harsher months. In contrast, the short sward of summer grazed pasture with Hereford cattle and rabbits seemingly eating plant life within an inch of its life may appear a desert of grass by comparison, but upon closer inspection, this herb-rich habitat is just of a different kind. Hairy and dog violets bloom above the short sward, providing food for violet cosmate moths, and cowslips can be seen on slender stems among the pats. To a Cowslip by James Hedderwick, abridged. No tiny alien plant art thou, but one to England's poet dear. The very pearl I witness now, he hung enchanted in thine ear. Mid mesh of matted grass and weed, O unchanged cowslip that I see, what time he strayed through Stratford mead, he saw and plucked a prize in thee. Now here, now there at his command, the lightfoot fairies come and go, while faintly from the silver strand the airy elfin trumpets blow. Thy lips with honeydew are fed, the glowworm lamps thy glades illume, ethereal banquets round thee spread, make endless sweet thy breath and bloom. O wizard wonder thus unfurled, ye take the grossness from the hour, with glimpses of a spirit world, through glamour of this golden flower.
So in this section of the video, I will be discussing the ways in which we measured species richness within two different areas of Durlston Country Park. Here is a list of things that I will be covering in this section. We chose fields 5 and 3 to compare to each other, as 5 was managed and 10 was grazed. There were many other differences in the fields too, like hedgerows and paths. We made sure that we started the study away from the paths, as they would likely affect the results. We would usually use a measuring wheel to mark out the area, but didn't have one available, so this part of the method took a bit of improvisation. We placed a bag at the starting point, then used the side of our 1 by 1 meter quadrat and flipped it continuously from there until we had gone up 20 meters. We then placed another bag and did the same again in another direction to create an L shape. This is how we made the axis. The unknown species we found were easily identified through an app called Google Lens. This app works for almost any object, and you use a phone camera to identify it. The app then shows the common and scientific name for the species as well as some information on it. We listed our species ABC in our results so that there was less information to write. This also avoided confusion and meant we didn't need to write as much in our notebooks. To get the numbers for our axis, we went round the group asking for a random number between either 1 and 20 or 20 and 40. This process was repeated until we had enough numbers for the 30 samples that we did. We used the same numbers in both of the fields. To find the numbers on the axis that the random number table gave us, we used large steps acting as meters. We used 2 people for this, 1 on each axis, and these people stayed the same so that the study remained consistent. 1 person read out the axis numbers and 2 people on them went to the right place and met in the middle. This is where the quadrat was placed. We all took part in counting the species in the quadrat so that it took less time and was more efficient. The results were written twice so that they were in more than one place. If there were any new species in the quadrat, they were identified before any of the others were counted so that they could be added to the key immediately. We found that the ABC method of listing species was very simple, yet very effective and neat. We repeated the whole process for the methods in both fields and did everything exactly the same.
In this part of the project, I, Harry Godding, will detail the results we gathered from our 2 sites at Delston Park and present them in tables and graphs to help understand them fully. On this slide, the first table seen at the top of the page details the species richness and abundance from Field 5 at Delston Park. From all 30 quadrats we sampled in this area, the highest species richness was 6 and the lowest 1. The highest species abundance was 62 and the lowest 4, with the total number of species observed in this area being 14. The average species richness of all 30 quadrats was 3.3 and the average species abundance was 20.9. Now, comparing this location to our second location, which the results for are detailed in the table at the bottom of the page, there are some key differences observed. Compared to the first site, this site was a grazed field, filled with cows rather than a meadow, so the species richness and abundance was a lot lower. The highest species richness recorded was 4 and the lowest 0. 2 of the quadrats in the grazed area contained 0 flower species, whereas none of the quadrats sampled in Field 5 had no species to sample. The species abundance of all flowers in Field 5 is also a lot higher than the grazed field as shown by the results, where the highest species abundance recorded in the grazed field was 20 compared to records of 62 from Field 5. The total number of species observed in the grazed field was only half that of Field 5 at 7, and the average species richness for all 30 quadrats was almost half that of the Field 5 area at 1.8 species per quadrat. The average species abundance from the grazed field area recorded was 6.3 per quadrat, which is more than 3 times less than the average abundance of species from Field 5 at 20.9. On this slide, 2 graphs can be seen, which show the cumulative mean of certain species of flower from our sites at Delston Park. The graph on the left details the cumulative mean of 2 rare species, the bush vetch found in our 30 quadrats in Field 5, indicated by the black line, and the speedwell found in our 30 quadrats in the grazed field, indicated by the red line. As seen on the graph, the bush vetch's line fluctuates until the 23rd quadrat, where it flattens at 0.7 until the final quadrat. The speedwell's line never stops fully fluctuating, but at the 15th quadrat it seemed to begin to flatten until an anomaly in the data called the line to spike, back up from the rested 1.2 to 1.7. The graph on the right showed the cumulative mean of 2 common species, one from each 30 quadrat at each location. The birdfoot found in Field 5 indicated by the black line, and the buttercup found in the grazed field indicated by the red line. The birdfoot's cumulative mean doesn't stop fluctuating even after 30 quadrats, so more samples may have to be taken in the future for a better result. The buttercup has a much straighter line, which could be because there is a lot less of them overall, but by quadrat 6 all results are within 1 of each other, and the line ends with the last 3 quadrats all stating as free as their mean.
In this section, I will be discussing the results in more detail as well as the significance of the results. The study hypothesis predicted that wildflower species richness will be greater in a meadow than in a summer grazed cow pasture. The results proved the hypothesis to be correct, with greater species richness found in the meadow than in the cow pasture. Despite this, the disparity in species richness between the 2 habitats was not as pronounced as was expected, with 14 different species found in the meadow compared to 8 in the cow pasture. Interestingly, the cow pasture also contains several species not found in the meadow habitat, with those species being cowslip, germander speedwell and crosswort. Such findings could be explained by the fact that the improved soil and/or lower vegetation of the cow pasture acts as a more optimal habitat for some species not found in the meadow, such as cowslip, which prefers habitat containing shorter vegetation in order to optimise sun exposure. In addition to this, some species may be less able competitors in the densely vegetated meadow habitat, allowing for greater propagation of such species in the cow pasture. Both factors explain the ecological characteristics of the cow pasture, which the meadow does not possess, something which may contribute to the finding of a surprisingly high species richness in the cow pasture. The higher than expected species richness and presence of unique species in the cow pasture reflects its ecological significance as a habitat for some wildflower species. For example, cowslip, which is also a species of conservation importance, with changes to British agricultural practices in the 1970s and 80s causing population declines. The results do, however, fundamentally support the hypothesis, showing that the maintenance of meadows is key to optimising overall wildflower species richness. Despite this, creating a habitat mosaic of meadows and cow pasture fields would be the optimal conservation policy, so as to enable the preservation of wildflower species not suited to meadow habitat. It is important to also acknowledge the potential limiting factors which may hinder the validity of the results. One such factor is the effects humans are likely to have upon the studied habitat. For example, despite carrying the study away from paths in order to minimise trampling potential, trampling may still have occurred on both fields, with one potentially suffering more than the other. The conservation practices carried out at Durlston likely also had an extraneous effect. For example, the meadow studies managed to ensure skylark nesting suitability. Such management practices may impact upon wildflower species richness in the meadow studied compared to other similar meadow habitat.
Recommendations. Is there any further useful information we could have done to make the field trip more successful? If we had a second opportunity for the field trip, what could we do differently? When considering how we identified the species of flower, perhaps we could have looked for a more accurate way to do so. When identifying species, we used the identification app iNaturalist. However, we encountered two issues here. One being the lack of sufficient internet service in the country park, therefore making it difficult to use the app, and number two, the risk of the species ID being incorrect. If we were to complete this fieldwork again, perhaps we could consider the use of a dichotomous key. This could allow us to determine the identity in the way of using a series of questions or characteristics to narrow down the species. Would this have been more helpful than using an app that we could not guarantee would be 100% accurate, as well as trying to access the internet on our phones in a location known to have bad coverage? Using a mobile phone application for species identity, such as iNaturalist, the app we used, cannot necessarily be a trusted or reliable source. The use of a dichotomous key would help to filter down to a more specific species of which you'd expect to be more precise. Moving on from identification, another improvement we potentially could make would be to measure the pH level of the soil. Had we borrowed more equipment on the day, we could have potentially measured the soil pH to look for a comparison between a field grazed by cattle and the relatively untouched meadow. Would the treading of the cattle on the ground affect the soil and therefore species abundance and diversity? According to the National Trust, cycles of grazing allow disturbed areas to be alongside areas that are rested and ungrazed, which gives a greater diversity of habitats. Furthermore, would knowing the grazing or cutting schedules of each field make a difference? Had we known when the grazing started or when the meadow was last cut, could affect our results. If we were to go back the same time next year, would there be a difference? This is likely, but why? Had we known these details and used them in our results, we could undertake the same study next year and compare those results. So could we have done more beforehand? Yes, perhaps we could have done more research on the park itself, its wildflowers, and its grazing and cutting schedules. It has since been interesting to find that Durlston have a daily diary that make notes of the weather, its wildlife such as birds and butterflies, as well as plants and flowers. Should we have spent more time looking at Durlston's website to gain more knowledge and information before we started? This certainly could have benefited us, but as the saying goes, hindsight is a wonderful thing.